Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and welcome to the live session 2 of Enzyme Science and Technology. So if you have any questions, you can ask me. Otherwise, I have brought some material that I can discuss with you, and hopefully this discussion will help you to prepare for your final exam. So first, I prefer that if there are questions, we can take the questions from you. If there will be no questions, then I will take it. I will going to discuss the content with you, and maybe that will refresh your memories about this particular course, and also it may probably bring some discussion or may bring some uh, it may actually bring some discussion, and you may actually be able to ask some questions also there. So I'll wait for 5 minutes in case you have any question. If you don't have any question, then we will start the presentation, and I will take you to the content what we have discussed so far, and then it will refresh and review your memories, and I hope that it will helpful for your for preparing for your exam also. So I request you that you should post your questions as soon as possible so that we may be able to do both. We may be able to discuss the content which I brought, and we may be able to discuss your questions. But your questions are our first priority. Uh, so please post your questions. Don't think about that there will be any question which is very silly and we may not, it may not be required discussion. We'll discuss each and every questions, even if they are as tiny and silly as well. So if you don't have any more question, I think I will share my screen, and I think we will discuss this material what I have brought. And then in the meantime, if you join and if you ask me questions, we will discuss. I'll be happy to answer those questions as well. So I'm sharing my screen. So I'm sure you can be able to see the screen. So this is the live session 2, and so far what we have discussed majority of the content. So we almost, I think, done with the week 8, and the so far what we have discussed many things related to enzymes. So you know that this course is devoted, or the focus of this course is about the enzyme, and we are not discussing the classical biochemistry or classical enzymology, where we were only talking about the enzymes and so on. I have tried to make it more practical oriented, so that it should be more useful for the people who are who are dealing with the enzymes, and then probably it may be help them to advance their work. And apart from that, since many of the MSc and as well as the BTech students will take this course, so I have also thought of putting the basic content. So I have put like enzyme classifications, enzyme nomenclatures, and so on. So overall, the purpose of this particular course is that it should actually give you a basic knowledge about the enzymes, and then at the same time, it may also should give you the I know the advanced knowledge, and as well as the theoretical practical knowledge, so that if you would like to be interested to pursue the content, or if you want to be pursue the research in this particular field, you should be well aware of the available literature. You should be well aware of the available techniques, so that it will help you in advancing your work, or it will make you life easy for understanding the difficult concepts. So the first thing what we discussed, if you recall, that what are the enzymes? So enzymes are also been called as the biological catalyst, right? And they are being called as the biological catalyst because they are present in the biological system, and the enzymes are present in all living cells, right? Whether it is a prokaryotic cell or a eukaryotic cell. Then what is the primary function of the enzyme? It's actually converting the substrate into the product, right? And so it is taking the substrate, and substrate is binding into the active site, and then at the active site, the substrate is going through the process of remodeling and those kind of things. So all the groups are going to be rearranged and all that, and then ultimately the substrate is going to be converted into product. And then once the product is being formed, then the product is going to be released from the active site. Mostly the proteins are, mostly the enzymes are made up of the protein, but there are an enzyme, but it does not have the proteinaceous structure. Apart from that, 99.9% enzymes are made up of proteins. And what the enzymes are doing, they are biological catalysts. So they are actually increasing the rate of the chemical reactions. But at the end of the reaction, they are remain unchanged, which means they are not going to be consumed within the reactions, right? And that's how they will remain within the reaction, and they will be keep increasing they will be keep taking the substrate and keep producing the products. Now, as far as the mechanism is concerned, how the enzyme works. So enzymes are the catalyst, right? They are biological catalyst, which change the rate of chemical reaction, but do not alter the mean. Is that suppose you have the two substrate A and B, and they are getting converted into C plus D, right? Now, they are initially there will be no there will be zero concentration of C and there will be zero concentration of D, right? That means that the equilibrium will be in this direction, right? And A and B are actually going to react with each other, and that's how they will be start synthesizing the C, right? So you imagine that A has OH and the B has a phosphate group, right? So in that case, what will happen is that the bond between the A and OH is going to be broken, and the bond between B and phosphate is going to be broken, and then the phosphate group is going to be transferred onto the A, and the OH is going to be transferred onto the B. And as a result, you are going to get the C, which is the A phosphate, and the B, which is BOH, right? Now, these are the products of C and D, which are being done by this particular enzyme. Now, what will happen is when the reaction will start in the beginning, there will be 100% A and 100% of B. But after this chemical rearrangement, and there will be 0% of C and 0% of D. But at the after, so the equilibrium or the reaction will run in this particular direction, right? And as a result, it is actually going to produce the C and D. So after some time, that when the C is going to be formed, then the same kind of rearrangement is also to occur at this particular site. And as a result, the reaction will run in the backward direction. And this will continue until they will reach to an equilibrium. And at the equilibrium, you will have some concentration of A, you are going to have some concentration of B, you are going to have some concentration of C, and some concentration of D. And this equilibrium is not going to be changed by the enzyme. What it is actually doing is it is only changing the forward directions, or it is actually changing the direction. It is only changing the rate of chemical reactions, right? Earlier, if you don't have an enzyme, the conversion of A plus B to C plus D would be very, very slow. But when you are going to have an enzyme, it is going to be faster, but it is not going to change the equilibrium concentrations. And how it is doing so? It is doing so because it is actually reducing the activation energy, right? So what it mean by activation energy is that suppose you have A and B, and it is actually forming a product of P and Q. So A and B are actually having these groups, right? OH and PO, right? Phosphate groups. And these exchange of material require them to transfer to cross through energy barrier, and that energy barrier is actually going to be very high if there is no enzyme present, right? So in an uncatalyzed reaction, the energy barrier is going to be on the higher side, whereas if the enzyme is present, it is actually going to reduce the energy barrier. That means it is actually allowing the easy crossing of the A plus B to form the P plus Q, and that is what the mechanism of how the enzyme is actually facilitating the process of conversion of A plus B to C plus D. So a catalyst function by lowering the activation energy of a reaction, the energy barrier for the reactants to become the product. Now, the question is why we need an enzyme, right? We need an enzyme because the enzymes are increasing the rate of reactions, right? Of reactions. Now, you see, I have given you an example of some of the enzymes, and here it is actually giving a reaction of the non-enzymatic reactions, and here it is actually going to say that what will be the rate of the reaction if the enzyme is present. So what you see here is that there will be an enhancement of 10 to power 6 or 10 to power 9 and 10 to power 11, 12, and 14. So this is actually a very high order rate enhancement, what you see when the enzyme is present into the reaction compared to the non-enzymatic reactions. Now, if you want to avoid the enzyme and if you want to achieve these reaction rates without enzyme, then what you are actually having an option. You are actually going to increase the temperature, so you can actually be able to operate these reactions at a high temperature, or you can actually be able to operate these reactions at high pressure, right? If you maintain the high pressure and high temperature, then probably you can be able to compensate for the loss of the reaction rate from the by not using the enzyme. But what will happen is that some of the substrate, right, are not going to be stable, right? So they will So substrates are not going to be stable if they are being kept into a high temperature and high pressure. For example, if I am keeping a, a high temperature like 120 degrees. So for example, if I am using the glucose and I want to convert that into glucose 6-phosphate, then with the enzyme, I can use the hexokinase, right? And I can be able to get the 6-phosphate. But if I don't want to use the hexokinase, then I have to use the high temperature. And if I use the high temperature like 120 degree or the 2 atmospheric pressure, uh, then what will happen is that the glucose is actually going to be decomposed, or it may actually going to lose its structure, and that is actually going to be a problem. Apart from that, these kind of high temperature could be possible in a test tube, right, in a reaction vessel, but it is not possible under the biological conditions. For example, biological condition, because most of the organisms are maintaining a constant temperature of 37 degrees Celsius, and they are also living in a constant pressure, for example, 1 atmospheric pressure, right? So these are the limitations, and under these limitations, you can there's only way you can be able to enhance the enzymatic reaction, that the rate of reaction, that you are actually going to have the enzyme into the reactions. Okay, and that is the easy way of doing it, and that's why the biological system has adopted the different types of enzyme for converting one substrate to another substrate, right, or one substrate into another product, and so on. So we discussed about the classifications. So there's a enzyme commission, and the enzymes are classified into 6 different groups, right? You have the all these 3 different groups, and this classification does not take the amino acid sequence, protein structure, or the chemical modifications. All enzymes are assigned as the enzyme commission number, right? And what is the enzyme commission number, and what information you will get? So enzyme commission is a very number which is given to be a particular enzyme. And what information you will get? You will get the complete detail of that particular enzyme. Let's see what information you will get when you have the enzyme commission number. So I have taken an example of the one number, that is 1111, right? So you actually, in the enzyme commission number, you are actually going to have the 4 numbers. So these are called as A, B, and C, and D. So here A corresponds to the class, or the such as the class 1, right? Then B is corresponding to the subclass. So in this case, the subclass is 1. Then the C is corresponding to the subclass, and that is the subclass 1. And the D is corresponding to the subclass, and that's how it is subclass 1. So if B and C actually give the information about the reactions, whereas D is the final number given to the particular enzyme based on the actual substrate into the reaction. So the EC number of 11111 says that the enzyme belongs to the oxidoreductase class. Okay, because oxidoreductase is the class 1, and it is belonging to that particular class. And within that class, you are going to have subclass, subclass, and subclass. So let's take an example, right? I got the EC number as 34114. Okay, so this is the EC number. So this is A, this is B, this is C, and this is D. So what is A? You have A, which is 3. You have a B, which is 4. You have a C, which is 11, and which you have a D, which is 4. So A is 3, right? This means the third class or third class. So what is the third class? Hydrolase, right? Now, within the hydrolase, you have a subgroup, and with this subgroup, you have a 4 number. So these are the subgroups, and this subgroup says that these are the hydrolases which are actually going to work on the peptide bond. Then you, within the subgroup, you are going to have subgroup, and SAS is actually going polypeptide. This means it is actually going to cut after a particular amino acid. And then within this, you are going to have a number 4, that is the D, right? And this is called as subclass. And these are the hydrolases which are actually going to act on the tripeptide. So these are the hydrolases which are going to work on the peptide which contains the 3 amino acids. Now combining this information, the enzyme is tripeptide aminopeptidase. You see that this C and D is actually going to give you the information about the substrate, right? So C is telling about that it is actually going to cut after the amino terminals, and the D is actually going to say that it is a protease which is actually going to work against the tripeptide. So based on this, the enzyme name is tripeptide aminopeptidase. Okay, and you can be able to get more information about this in a XPASI. So then we have also discussed about the classifications, rules of classifications, and rules of nomenclature. So the rules of nomenclature: generally accepted trivial name of substrate may be used in the enzyme name, for example, the pepsin and all that. Then rule 2, where the substrate is a normal in the form of an anion, name should end in the ate rather than ic, right? For example, the lactate dehydrogenase, the lactic dehydrogenase or the lactic acid dehydrogenase. Then rule 3 is commonly used abbreviation for substrates such as the ATP may be used in the name of enzyme, but the use of new abbreviation should be discouraged. So these ATP and DNA, RNA, these are very, very well accepted abbreviation, but there are abbreviations which are not very common and which are not very popular. So those abbreviations are not allowed. And then rule 4, the name of the substrate composed of two nouns should be hyphened when they are forming part of an enzyme. For example, glucose and the phosphate, right? So you say glucose 6-phosphate 1-dehydrogenase, okay? Then rule 5, the use of enzyme name of description such as condensing enzyme, pH of enzyme should be discontinued as soon as the catalyzed reaction is known, okay? So this also is very, very discouraged. Then rule 6, where the true acceptor is unknown and the oxidoreductase has only been shown to react with the artificial acceptor, the word acceptor should be written in the parentheses, as in the case of the succinate acceptor oxidoreductase. Then rule 7, the systemic name should have two parts, name of the substrate and the process specifying ending with the ase. Then we also discuss about the enzyme classification. This is the simple example of the oxidoreductase. Oxidoreductase is belonging to EC1. And when we were discussing about the enzyme classification, we discussed in detail about the all the classes and what reactions they are catalyzing and what kind of how you can be able to, what are their requirements and so on. So for example, in the case of oxidoreductase, it catalyzes the oxidation reaction, which means it is actually going to have the transfer of hydrogen or oxygen atom and electron from the one substrate to another. This means AH plus B is actually going to give you the A plus BH, this is a reduction reaction. Similarly, if you AO, this NADH or FADH. Some cases you are also going to have the NADPH. NADPH is mostly being present in the plant oxidoreductase enzymes. And the examples are the dehydrogenases and the oxidases. For example, the lactate dehydrogenase, alcohol oxidase, both of these enzymes are part of the anaerobic oxidations. So this is just a simple example. We have also discussed about the other classes like the hydrolases, isomerases, and also on. So this is just an elaboration of the oxidoreductase, and we discussed all the reaction mechanism and all other things. And then we also discussed about the enzyme structure. So we discussed about the primary structure, secondary structure, tertiary structure, and quaternary structures. We discussed about how you can be able to determine the primary structure, so you can actually be able to do the protein sequencing. Then we discuss about the secondary structure. So for the secondary structure, you can use the CD and IR. And I have taken to my lab also to you, and where the Professor Nitin Chaudhary has discussed how you can be able to collect the CD data and how you can be able to analyze the CD's data. And then when we were discussing about the tertiary and quaternary structures, we discussed about how you can be able to determine the tertiary structure and quaternary structures, and what are the different methods are available and how you can be able to approach. And in that process, we have demo videos so that you can be able to use them for your advancing your work. So, for example, when we were discussing about the determination of the protein sequencing, like the primary method, primary structure, we discussed the detail of the Sanger's method and as well as the Edmond degradation methods. And what we said that if you have a protein, you can actually be able to first reduce the protein with the help of a you reduce the disulfide bonds so that it is actually going to have the linear chain, and then you are actually going to generate the different fragments with the help of the trypsin, and then you are going to do a sequencing by the Edmond degradation method. So when you do the cleavage by the trypsin, you're going to generate the different fragments, right? And then these fragments are actually, each fragment is actually going to sequenced by the Edmond degradation method or Sanger's method. And then they are actually going to placed by establishing a sequence, right? You can actually be able to use the overlapping sequences, and that's how you can be able to get the final sequence. And then we also discuss about the tertiary and the quaternary structure. So we discuss about the molecular modeling. We discuss about how you can be able to use the modeler, right? And we also discuss about how you can be able to do the molecular modeling with the help of the homology modeling and all other kind of things. And then in detail, we discuss about what are the different, methods you can be able to use, what are different softwares are available, what are different approaches you can use, and so on. And then we would also discussing about the experimental method. So in the experimental method, we discuss about the two method, that is the crystallography and as well as the NMR spectroscopy. So in detail, I have shown you how you can be able to do the crystallization, and I have actually given you a demo also that how you can be able to set up the reactions, how you can be able to monitor the crystal formation and so on. And then subsequently, we have also shown you how you can be able to collect the diffraction data, how you can be able to analyze the diffraction data, and how you can be able to solve the structures. Same is true when we were discussing about the NMR spectroscopy. So we discussed very briefly about how you can be able to do the NMR spectroscopy and so on. And ultimately, you're going to get the tertiary structures. Then within the homology modeling, we discuss about the step, all these steps. So we have a 5 steps process of doing the homology modeling. We have shown you a demo also, and we also, in fact, given you a demo about the protein structure analysis also. So you can be able to do the protein structure analysis, and using this protein structure analysis, you can be able to get a detailed insight of the protein, what will be the function of this particular protein, and what will be the role of this protein in catalyzing a particular reaction, and what part of the protein is actually participating into the these kind of the accepting the substrate or converting the substrate, what are crucial amino acids are present, and so on. So this is in detail actually we discussed, and I have given you a link also that in case you want to read more about these molecular modeling, you can be able to go through this particular the article, and it will actually going to give you in detail about how you can be able to do the molecular modeling. And then we also discuss because all these processes can only be done if you are actually going to have an enzyme. So we also discuss about how you can be able to clone the enzyme, how you can be able to do the overexpression, how you can be able to do the purification, and so on. And in detail, actually, we discuss about how you can be able to identify a gene which is for the particular enzyme. So we discuss about how you can be able to do when the genomics are not known, or how you can be able to do if you don't know about the genomic sequences and it's only the cDNA sequences are known, and so on. And then we also discuss about the cloning. So we discuss about the gene amplifications, cloning, screening, and then we were discussing about the overexpression. We discuss about the host. We discuss about the different types of host, like the prokaryotic host or the mammalian host or the yeast, Z cells. We discuss about the DNA delivery, screening, overexpression, and analysis. And for majority of these part, we have actually given you a lot of demo videos so that you can be able to get real, experience of the how you can be able to perform these. And then we also discuss about the purification. So we discuss about the cell disruption, chromatography, then we estimations and purification analysis, so on. So till then, I think this is the place where actually we have actually stopped, and in we are actually going to cover some more content related to like how you can be able to study the enzyme kinetics, how you can be able to develop the inhibitors, and how you can be able to develop study the inhibitor kinetics, how you can be able to identify the mode of mechanism of enzyme inhibition and so on in our subsequent videos. So till then, I think if you have any questions, we can actually be able to take those questions, and I'll be happy to discuss those questions. And I have refreshed your memories. The purpose of this live session was that I should be able to refresh your memories, whatever we have discussed so far, so that it will help you to prepare for your exams, which are only the next month only. And if you have any questions, please post your question into the chat box so that I'll be happy to answer those questions. Otherwise, this live session is very limited, and if there'll be no question, I think we'll wind up this even earlier also. So with this, I think I would like to come out from this presentation. And if you have any question, please feel free to drop your message into the message box, and I'll be happy to and discuss those points. So So I think you don't have any more questions, and we have discussed in detail about the content what we have discussed so far in the course. And I hope that discussion could be useful for you to prepare for your final exams. If you have any more questions, any more queries, you can actually be able to drop your messages into the discussion forum, and I'll be very happy to address those queries also. So with this, I would like to wind up my live session for today. And if you have any more questions, please don't hesitate. You just drop the questions and queries, and we'll be happy to address those queries. Thank you.